Okay, for the next talk, we're going to invite Amir Hossein Dakhili. He's a PhD researcher focusing on the brain mechanism underlying cannabis craving. He's using functional magnetic resonance imaging at 3D and 7D scanners to investigate how different brain regions active and communicate with each other, while the individuals with cannabis use disorders are exported to cannabis stimulants in order to identify specific neural pathways associated with craving. Furthermore, he's exploring the use of real-time function MRI neurofeedback to modulate this brain response to enhance our understanding of causal link between specific brain activation and cannabis craving, and ultimately inform the development of novel intervention for cannabis use disorder. Let's welcome Amir.
Hello, everyone. Thanks so much for attending the educational course today. Of course, that it was engaging enough until now, and what I'm going to do is that I'm going to go into more details of whatever Chao and Sampras already talked about the application of real-time neurofeedback. So we remember that the story begins with the scanner when it sends the data and Turbo Brain Voyager in our case, or any other real-time processing software. We receive this data. It will analyze this data in real time and pass it to the front end, which is the MATLAB or any other programming script that wants to result in showing that feedback, that data to the participant in real time. So whatever you see here, if it's going to be a continuous feedback, which it is in our case, should ideally repeat every 1 second. This loop should ideally repeat every 1 second and result in a updated feedback showing to the participant. We'll start with receiving data from the scanner. And next step would be Turbo Brain Voyager. So Chao and Sampras mentioned Turbo Brain Voyager before, and it is also one of the tools that you can use for real-time data processing. I highly recommend throughout the talk, if you can scan any of these QR codes, it will be beneficial because we also have like a different GitHub repository for any like codes that you might need to use for generating any task-related or experiment-related data that you need. And also, this one specifically is for Turbo Brain Voyager. I highly recommend just go through the website, download the documentation, and whatever you see today will definitely help you to get a better sense of how Turbo Brain Voyager actually works. But I can say that it is user-friendly software. Also, you need to always think about how can you actually use documentation to learn what exactly are you looking for and select the right choices for your experiment. The first thing we need to do after we receive data from the scanner in the first part before starting the actual neurofeedback or brain training task. So I'm going to separate it in 3 different steps. One is going to be receiving data for the structural images. Then I will describe what we will do in localizer run that Chao and Sampras already mentioned. And then I will go through the actual neurofeedback. So this is what happens before the neurofeedback session itself. So what happens is that a scanner will send a structural data, which might take about 7 minutes. And after that you have this structural data, you will receive it in Turbo Brain Voyager. And now is the time that you need to make the structural mask for participants. So as you can see here, you receive all these DICOMs that came from the scanner. And there are different steps that you need to follow in Turbo Brain Voyager to first shape the 3D brain model in Turbo Brain Voyager. After that, extract the brain itself and kind of correct any inhomogeneity in the brain. And also make it as a MNI standard template. Because it's important, you want to receive activation in real time, and you want to put that ROI mask on the brain. You've already defined your ROI mask based on MNI template. So it's important to have all your structural data of every participant based on a MNI template. So as you can see here, brain has been extracted, and the last step would be to kind of generate the MNI template of the structural data, which all these steps will be followed in Turbo Brain Voyager through the menu. Again, for any details of these steps, to keep it in mind, use the documentation of Turbo Brain Voyager. And as you can see, this data is now is MNI standard and ready to be used. Okay, now we are going to the localizer run. What's happening in the localizer run? Just as a review. The idea is that, in our case, we already defined a mask, which was ACC, anterior cingulate cortex. But we don't limit ourselves to anterior cingulate cortex, and we want to see for every individual which specific voxels within anterior cingulate will be more activated. So this is the reason that we are using the localizer task. As Chao mentioned, in our case, localizer task is a Q-reactivity task. We have a couple of images related to cannabis or neutral images. What participant needs to do is only look at these images and pay attention to these images in the real time. Data will be transferred to Turbo Brain Voyager. The mask of that predefined ROI is already in there. And what we need to do is to extract those most activated voxels within ACC. So, as I said, there's going to be a real-time data processing here. We still didn't start the actual neurofeedback. It is just a localizer run. We have the initial ROI mask. And the other thing that we need is a protocol file. So again, you see the QR code, and you can scan, and you will have a code that you can generate this protocol file. Where does it come from, and what does it include? Protocol file has the timing of every block in that Q-reactivity task. You need to define for Turbo Brain Voyager that what's happening inside. You're saying that, okay, I have, let's say, 400 volumes, but you need to also mention that within these 400 volumes, from what second to what second is related to cannabis cues or neutral cues. All these data of onset and offsets of every cue is within the protocol file that you can generate based on the code and based on the output that you receive of your code. So maybe before the day of the scan, you will run the code for yourself. You get the output of timing, you use the code, generate the protocol file. Then you have all this timing of onset and offset, and you give it to the Turbo Brain Voyager. The first thing that you see here is an example of the target ROI mask. When we say target ROI mask, we are talking about the coordinates of the mask. So they're going to be a text file, and you use this text file as an input for Turbo Brain Voyager. The second one is the protocol file that I mentioned. Onset and offsets of every cue, that in our case was cannabis and neutral cues. And just as a review, data comes from the scanner and goes to the Turbo Brain Voyager for real-time processing for these Q-reactivity tasks. A couple of important steps before starting the Q-reactivity task is how to actually set it up in Turbo Brain Voyager. There are lots of information about it that I cannot definitely cover it today, but I highly recommend that you go through documentation again. One of the important things in this setup is timeout seconds, as you can see here. This is where we define, please wait for these couple of seconds if you didn't receive any data from the scanner. Just keep in mind that it's a real-time processing. If there are going to be any issues with the connection between a scanner and Turbo Brain Voyager, and TurboBrain Voyager doesn't receive this data, it might crash. So we define these timeout seconds here to say that, okay, please wait for these couple of seconds before crashing, data will come. Or if not, then feel free to crash, at least after these couple of seconds. The other thing that is important is, you see here 3 different boxes. The first one is called watch folder. Watch folder is the folder that TurboBrain Voyager looks into and waits for those DICOMs that are coming from the scanner. The second one is a target folder. These 2 are mostly important for neurofeedback. So let me just leave it for the part that I'm going to describe neurofeedback, but briefly I can say that these 2 are those folders that the output of TurboBrain Voyager will be saved to. So it's also important to keep them in the right direction. The other thing that you see here is you need to define the exact number of DICOM series that are coming from the scanner. Even before query activity task, you had that structural data that came through. You had any, maybe like phase encoding scans that are coming through, and all of these has a specific DICOM series number assigned to. When you want to ask TurboBrain Voyager that now I want to start query activity task, you should also define that this is the exact number that you should wait for as DICOM series. So it's also important. And the other thing is that you should keep in mind to kind of upload this protocol file and also the number of volumes that you have within this specific task, which is important, and your TR. And lastly, one of the most important things is that based on the files that you've generated as the structural templates, you need to upload all those structural images resulting from each participant's structural data here in the last step of this setup. So you remember that we had a back-end, front-end. Back-end is what you see, as Chao mentioned, in the MRI control room. And front-end is what participants is, but even in the background of this front-end, there's a script that is running in the real time. So what's happening here, if you look at to the left side of the screen, you see that blue part, that's actually our predefined ROI, which was the ACC. At the same time, like this is the time that we start scanning TurboBrain Voyager and the task at the same time. So this is what actually happens. Participant looks at this task, and in the real time, you see that there are going to be some change in activity in the brain, and we are now want to focus only on the ACC and some mostly activated voxels within ACC. So here, what we'll do after the end of this query activity task is that we need to go through the VUI function to extract those specific voxels for each participant that were mostly activated. So basically, the output of this file will be, again, a number of coordinates related to those specific personalized voxels within ACC that we need to save within another text file and make it ready for the neurofeedback session. Basically, this will be those voxels that data for neurofeedback will come from. So as you can see here, after the query activity task, we highlight our VUI, select ROI to VUI to convert it to ROI, and adjust these VUIs based on the functional and activation that we had in the brain, and save this data in another .roi file to be used for the actual neurofeedback experiment. Even the order of what you see here is so important to kind of build a .roi file from your VUIs. So as you can see here, there are those yellow dots within this background of your ACC that are, let's say, the personalized voxels within ACC for each participant. that will be the place that this change in activation for neurofeedback will come from. But this data might be noisy, correct? We need to also think about and pay attention to any sort of physiological noise. So for this, we also define a confound region. A confound region can come from different resources and based on different literature, also the work that has been done by Sampras. We used an overlap of different confounds, which was, in our case, mostly the CSF. So we want to say, now I want to have my target ROI, which was that personalized ROI, and I also want to have that data from the activity within that confound region. And I want to use this as noise. So at some stage, not in TurboBrain Voyager, but in my MATLAB script, and right before showing to participant, I want to regress out this data from this confound region, just to make sure whatever I'm presenting to participant is the pure activity of that target region, not any noise. So this was about the confound region. We want to remove physiological noise, and we need to regress it out at some stage to have a pure data of that target region. Let's go to our experiment. Chow already mentioned this is a complicated kind of design of our task, but just to give you a better sense, it contains 4 different runs. And in 2 of these 4 different runs, we ask participants try to upregulate your craving. Even I can say that, as you can see here, for example, let's focus on downregulation, which is the upper one, as you can see. This first blue block that you see here is kind of the first baseline condition that we have. This first baseline condition is a bit longer. It gives TurboBrain Voyager a proper time to find a baseline activation. Because keep in mind, whatever Brain Voyager wants to report us is going to be a % signal change of activity within that target region. So TurboBrain Voyager needs to always say that this change comes with respect to a baseline, how much it changed with respect to that baseline. This is the baseline that we define, and it grows over time during the task. There are also different other rest periods between our regulation conditions. So in each run, we have, let's say, 5 regulation blocks that we ask participants to either upregulate or downregulate their brain activity. And again, as you can see here, there's going to be a bar on the side of that image that they see during regulation phases, that they can see how successfully they could kind of up or down regulate their brain activity. And of course, that subjective craving questionnaire or fast questionnaire. Okay, we now have the target ROI and confound ROI. Everything is kind of ready to start the neurofeedback experiment. Again, keep in mind, timeout second is important. This watch folder was the folder that waits for the diagrams from the scanner. But now let's focus on this target or feedback folder. So target folder gives you general reports of what's happening and what happened in TurboBrain Voyager. But you remember that we said your script needs to receive data from TurboBrain Voyager to calculate the feedback and give it to the participants. The data that your script needs to receive every TR, which in our case is every 1 second, is the data that will go into this feedback folder. These files of data, they are called RTP files. What they include? They include activations, % signal change of your target region and your confound region separately. And it's the art of your task, your script, that needs to regress out the activation and the person signal change, not activation, person signal change of your confound region from your target region. So you need to keep in mind to select the right folder for your RTP file. If it's, for example, the first run, you probably have a folder named NFB RTP1. And your script will look for these RTPs to come into NFB RTP1. If you define another folder, your script can't find it. It results in your code crash. So, again, DICOM series is important. Localizer run is finished. You need to update this DICOM series. And a couple of other important things about the number of volumes and the protocol file. That, again, you need to have that protocol file here as well, like the timings of the actual conditions within your task, the timing of the blocks, your regulation blocks, your neutral blocks. TurboBrain Voyager needs to know about these timings. And the actual templates of structural images is also important. So what you see here is that the things that we already mentioned about neurofeedback, after you set up the neurofeedback and TBV, you also preload your ROI mask, which in this case is going to be not only the target region, but also the confound region, because you need to extract data of person signal change of both of these different regions. And after that, there is also this option in TurboBrain Voyager that you can define what is the threshold of your person signal change that you want to report. Or, for example, you might also want to ask TurboBrain Voyager that even within these most activated voxels that I defined, just give me the person signal change of most 33% of mostly activated voxels. So you can just narrow it down, and it's the option that you can use in TurboBrain Voyager. And as you can see, this was the structure of your ROI file. So there are these coordinates of both target region and confound region that you give it to the TurboBrain Voyager. And there are also different options in TurboBrain Voyager available. For example, you might want to just save the data from any processing timing, any delay of timing that might be important for you. You can just record all these delays of this real-time processing data and just keep it somewhere, store it somewhere at the end, and probably you can come with a report of this as well. That, for example, here you see that in a couple of pilot studies, as Chow mentioned, over 14 participants, we could find that, for example, there were some fluctuations in processing timing. Let's say starting from 500 milliseconds to even 2 seconds was the actual timing that it took from TurboBrain Voyager to analyze this data in real time. But the average of this processing time was about 900 milliseconds, which was a bit less than our TR and is kind of acceptable. I can't necessarily say successful or not, but less delay is always better. So, as you can see here, a couple of important notes. If I go back to the slide, probably it was to kind of just look at the video again. So we set it up, and the other thing that might be important in this case is that we actually need to activate to extract this RTP data, because one of the errors that might happen is that you set everything, but you don't ask TBV to give me those RTP outputs. So if you don't activate this option, there won't be any outputs as well. So this might also result in crashing your code. And this was those sort of troubleshooting or any reasons behind doing all these pilot scans again and again, because you need to have a good checklist of what you need to do just setting all these parameters based on the needs of your specific experiment is so important. So keep in mind to kind of activate any option that you need. So yeah, just briefly check extract time logs, which was the delay of processing if you need to. Don't forget to kind of upload your ROI. Again, it is important. If you don't define the ROI, you won't receive those data from your ROI. So all of these are important, and the protocol timing file as well, which is so important. And in your neurofeedback script, so this is the task that you want to kind of show the task to the participants. And what happens in your script is that, again, first of all, this task will show those images to the participant. It has a bar on the side that shows the feedback to the participant in terms of presentation. The other thing that it does is that it wants to access those generated data from TurboBrain Voyager, which was the RTP files, and data within this RTP file, which was related to target or your confound region. And it actually wants to regress out data from your confound region and calculate the feedback, present the feedback, for example, in shape of a thermometer to the participant in our case. So they can see that if the thermometer goes up, it means like a higher craving. They could upregulate their craving or downregulate their craving if it comes down. Yeah, as I said before, feedback calculation is probably one of the most important tasks for your script before showing to participant. And in our case, the other thing that we did that was that we used a sliding window for calculating this feedback, because one thing that is important is that these values of RTPs are coming every 1 second. It might be a bit jumpy. We found it jumpy where in so many of these pilot studies. If you want to present this continuous feedback every 1 second based on the data of only that 1 second, we found it's a bit jumpy. The thermometer just goes up and down, and it might result in a distraction for participants. So we kind of try to make it a weighted feedback, which we gave weight to the feedback not only from the last person signal change. By person signal change, we mean how much actually the activation within those specific voxels changed. We gave weight to the last second and 3 previous person signal change as well, to kind of try to normalize the feedback that we are presenting to the participant. Again, back-end, front-end. You already remember that back-end is the part that we see as researchers in the room, and front-end is actually what participants see in the scanner. So yeah, what's happening here is that you have your TurboBrain Voyager. You receive this data from the scanner. TurboBrain Voyager is working at the same time with all those protocol files and everything that you defined. And it will all result in showing these tasks to the participant, and they can see if they could actually upregulate or downregulate their craving while they're in the scanner with all those things that are happening in the background. Now you have a better view of what's happening, and then after each regulation, there is this neutral condition that they just simply look at the images, they don't need to change anything, and after this neutral condition, there will be another regulation block. Every run for us has the same regulation, only up or only down. But here, I just wanted to give you a better sense of how downregulation might look like. So in a separate run, their task will be to kind of downregulate their craving. And again, the same thing happens in TurboBrain Voyager, and they can see if they could successfully downregulate activation in those 33% of most activated voxels in ACC, which was kind of individualized for every participant. But as you remember, to reach these thermometer and kind of test your script, you need to have this data come from the scanner, and then you need to have Turbo Brain Voyager to analyze this data. But if you want to test your script when you're just checking everything is working, probably you can have, again, like if you scan this QR code, you can have a kind of another script. So probably you have a bunch of RTPs somewhere else in another folder. Using this script, you can ask your script to kind of drop your RTPs into a new folder. And instead of having all that set up in the background, you can just drop RTPs every 1 second into a new folder, which you can use it as a watch folder for your script. So again, you can get the same results, and you can see that your task will find those RTPs somewhere and can kind of try to show if the thermometer is working and if it goes up or down. So as a dry run, you can use this code to kind of access these RTPs in real time. Okay, and after this analysis, this is gonna be a whole different story, but I'm just briefly going through if you needed to know more about it. My poster will be on Friday and Saturday. We can have a chat about the results of this pilot study and what we actually got and how to download data, make it as a bits format, and then after that, what measurements we use in FMRI prep for pre-processing, and then even for spatial smoothing that are also important. We also did a regression analysis that in regression analysis, we wanted to kind of use it as a proof of concept, like whatever we are saying that this data is coming from anterior cingulate cortex and this person signal change. What if we define every person signal change as a regressor? And then we give it to the data as a post-hoc analysis. Does this pattern of regulation that we saw as person signal change, can it actually predict the activation within ACC? Or it might also result in predicting activation in other parts of the brain. And the results were actually interesting. This is only for one participant, but even as a group level, you can see that we used personalized ACC, confound region, anatomical ACC, which was that bigger region, and right hand knob as a control region, as somewhere random, to see whether there's going to be a pattern of a specificity of the feedback that we provided to participants. And as you can see here, those regressors as a prediction of the pattern of activation could show that, yes, actually we could see that those specific voxels that we defined for every participant had the highest correlation with that pattern of activation. So far we got these results, and just lastly and shortly, we could find a kind of pattern of learning as well, like a more consistent activation. If you look, run 1 and run 3 in our case was upregulation. Run 2 and run 4 was downregulation. We see from run 1 to run 3, there was a bit of less noise. If you remember, this was only a prediction based on those regressors. And we see that they probably showed a more consistent pattern of activation within ACC in round 3 comparing to round 1. And even in round 4, comparing to round 2, we see even more higher correlation in ACC and, of course, some other parts of the brain. I want to thank all the team behind this from both Australian Catholic University and Melbourne Uni, that it was impossible without their help, and also Australian government research training, a scholarship in National Health and Medical Research Council. And yeah, feel free to scan any of these codes to stay updated with any results or upcoming findings of this project. And thanks everyone for listening.